Hello, welcome to 13 Universal Learning Beyond Borders. This is Shedro, your biology tutor. So we are going to be solving questions on A-level CIA biology. I want to say that all the materials, the questions we're going to be using in this video, they all came from CIA past questions. Okay, so that's where we got them from, right? And therefore preparing students for the exam. So once you learn this, you'll be able to solve more questions. Okay, so just come with me. In this biology session, we are going to be looking at cell structure series 4. We have done series 1, series 2, 3. Please get the videos on 1, 2, 3 and then watch it. You're going to learn a lot. Very educative. So right now we are going on the first question. So which role matches each organelle with its function? Okay, yeah. So which role matches each organelle? Okay, let's eliminate the microtubules. Let's go down. So microtubules make cilia and spindle. Okay, well, yes, part of cilia and spindle from the basal membrane. Okay, of cilia, and then when they organize, they form spindle. So this is correct. Okay, so if I go to the B on microtubules, let me just eliminate, move vesicles within the cells. Yes, microtubules can do that. They form part of cytoskeleton. C says move chromatids apart during anaphase. Yes, they do that. Anaphase of mitosis, they help to separate them, right? They shorten at the poles and then pull the sister chromatids apart. Okay, forms part of cytoskeleton. Yes, of course, said that. All right, so any of these options for microtubules is correct. Let's go to rough endoplasm. Let's eliminate. It says separate some processes from cytoplasm. This is wrong. So we eliminate A because of this. This is not part of the work of endoplasm reticulum. Assembles amino acid to make protein. Now this can happen in rough endoplasm reticulum because of the presence of ribosome there. Okay. All right. So ribosome allows this to happen. Okay. Transport, and of course we'll come to C, modifies protein which may be released. Yes, rough endoplasmic reticulum may modify proteins. However, we know that Golgi apparatus is in charge of modification, but rough endoplasmic reticulum can also modify proteins, make triglycerides. They cannot make triglycerides. So we have eliminated D and A and D. So we are focusing on these two. So let's go to Golgi. So it contains enzymes for destroying one out part of the cell. This is actually made for lysosome. Lysosome contains this. Though lysosomes are formed from Golgi, but their sole function is this. They contains enzymes for destroying. Okay, so I think this eliminates B. So if you check this says makes glycoproteins. Yes, Golgi makes glycoprotein. Golgi makes glycoprotein, all right, by a process called glycosylation. Okay, there's a process called glycosylation. I don't know what I've heard about it. Glycosylation. So glycosylation, this is spelling here. Glycosylation is a process of adding, you're adding glycosylation. Let me wipe this. Okay, glycosylation. So it's the process of adding glucose to a molecule, glycosylation. Is that okay? So look at it. So Golgi can add glucose to protein to form glycoprotein. It can as well add glucose to lipid to form glycolipid. That's part of the modification process of Golgi. So guys, if you look at this, you see that this is correct. And of course, centros move cilia, right? So C is the answer to this question. The answer is C. Pause this video or watch it again and send us your comments. What do you think? All right, send us okay, please like the video, also share with your friends. All right, so let's go to the next question. It says, where are ATS ribosomes found? No, they are not found in chloroplasts. Okay, chloroplasts contains 70 70s ribosome, which is a smaller ribosome. Cytoplasm of eukaryotes, no. Okay, yes, they are found in cytoplasm of eukaryotes. Cytoplasm of eukaryotes contains 80S ribosome. Okay, so 2 is correct. 3 is mitochondria contains 70S ribosome, just like chloroplast and prokaryotes. Cytoplasm of prokaryotes, no. This number 1 chloroplast, 3 mitochondria, and cytoplasm of prokaryotes, they contain 70S ribosome, not 80. So what is the answer? Okay, so the answer is not A, is not B, is C, Charlie. Okay, because that is where we find 80S. So the answer is C, correct. Let's go to the next question. So which feature, when present in an organism, enables it to be identified as a prokaryote? So let's see features that enables to be identified as prokaryote. So cytoplasmic DNA. Well, yes. Okay, now the DNA of prokaryotes lie, okay, if you remember, the DNA lie, okay, on the lie in the cytoplasm, so they are not surrounded by what we call nuclear membrane. So for example, pardon my drawing, yeah, so let's assume this is a prokaryote. Of course, pardon my drawing, I'm using pen here. Okay, now their DNA lies, okay, freely on the cytoplasm, and this portion where it lies is called nucleoid. Okay, so that region is called nucleoid. So they are the only ones, prokaryotes, have cytoplasmic DNA, but they don't have nuclear DNA because their DNA is not enclosed in the nuclear membrane. Okay, so yes, this can identify, there is, this is correct, cell wall. Well, cell wall may not be a way to identify prokaryotes because eukaryotes have cell wall, plants have cell wall, prototists have cell wall, fungi have cell wall. Okay, nucleus, definitely they don't have nucleus, all right, because their DNA is not enclosed within nuclear membrane. They don't have definite nuclear membrane, all right? So you can't use it to identify them. Ribosomes, you may not be able to because they have 70S ribosome and 80S, and those ribosomes can as well be found in eukaryotes. The only thing that can identify them, all right, is cytoplasmic DNA discretely from other organisms like eukaryotic cells. So the answer, of course, is A. Let's go to number 4. What is the function of nucleolus? The formation and breakdown of nuclear envelope. What do you think? Pause the video and check it. No, A is not correct. B says formation of rough endoplasmic reticulum. No, rough is formed from nuclear membrane. Rough endoplasmic reticulum is an extension of nuclear membrane. So nuclear membrane is continuous with rough endoplasmic reticulum. So B is wrong. The synthesis of ribosomal protein, well, it does not synthesize protein. Protein is made in the ribosomes, not in the nucleolus. So D, synthesis of ribosomal RNA. Yeah, that's correct. Yeah, ribosomal RNA is made, all right, in the nucleus because nucleolus is a site for synthesis of ribosome. Okay, so D is our answer. Let's go to number 5. Which eukaryotic cell structures all contain nucleic acid? Wow, that's interesting. Cytoplasm contains nucleic acid. Messenger RNA is found there. All right, moving from nucleus. All right, transfer RNA is found in cytoplasm. All right, okay. Of course, in prokaryotic cells, DNA can be found here because the DNA lies. They have cytoplasmic DNA. Now, Golgi bodies doesn't have nucleic acid. So that disqualifies A, eliminates it. So central doesn't have nucleic acid, they have protein, tubulin protein. Okay, so that eliminates B. C says central again doesn't have nucleic acid. Okay, that cancels C. So the answer, let's look at D. D says chloroplast. Yeah, chloroplast has circular DNA. Of course, it will have messenger RNA, transfer RNA, ribosome RNA, has 70S ribosome, as ribosome has ribosome RNA. So the same thing with mitochondria, circular DNA, it will have, and it also has 70S ribosomes, so it will have messenger RNA. All right, they have nucleic acid. Cytoplasm also have transfer RNA, messenger RNA, Okay, ribosome has messenger RNA and ribosomal RNA. So all of them have nucleic acid. So the answer is what? D. Okay, please want you to like this video, share this video, and ask question. Okay, it's a which statement about viruses are correct? Which statements? Let's look at them. How many statements? Okay, 3. So pause video and check which one. They contain DNA or RNA. Yes, this is correct. DNA or RNA. Okay, DNA or RNA, not both. DNA or RNA. They use host cells to synthesize virus proteins. Yeah, this is correct because they lack organelles. Virus lack organelles, right? So that's why they are said to be acellular. They lack cell organelle, and of course, 3 says they can have protective coats of peptidoglycan. This is not correct. Viruses don't have peptidoglycan as protective coats. What they have is capsid protein coats, capsid, not peptidoglycan. Peptidoglycan is found in prokaryotes, not in virus. So 3 is wrong. So which option is correct? Is it B? Whatever there's 3 is wrong. A has 3 eliminated. C has 3. So the only one that doesn't have 3 is B. So the answer is what? B, of course. Let's go to 7. What are the features of prokaryote cells? Chloroplast and mitochondria. Okay, let's look at it. Yeah, this is very important. So the first one says circular DNA. They all have circular DNA surrounded by double membrane. Well, only chloroplast and mitochondria are surrounded by double membrane. So this is not part of it. Ribosomes smaller than 80S. Ribosomes smaller than 80S. Okay, that's 70S ribosome. Yeah, 3 of them have 70S ribosome. So they have both this, and the intercept is B. So B is the answer. Okay. All right, guys. Please, I want to thank you for watching this video. Please don't forget to like the video, and don't forget to share it, make a comment. See you in series 5. We are done with series 4 of cell structure. Bye. This is Shedro. See you next time.